instead. All right. But what happens now if I make an F2 generation? So if I cross these to each other, okay. So first of all, what colors am I going to get out, and in what ratio? Okay. And so the bottom line is it's actually still going to be a simple Mendelian pattern, but this is how it's going to turn out. So now you get sperm having either CR, CW, eggs being CR, CW, the same Punnett square. Okay. But now you've got 1/4 are red, 1/4 are white, and 1/2 are pink. Okay. That's pretty straightforward. Okay. But now what you see is that the phenotypic ratio is the same as the genotypic ratio. Okay, and that's because these two, so in this case it would be, well, the equivalent of big A and big A little a, they don't give the same phenotype. Okay, so instead of getting a 3/4 here, again, you still have 1/4 red, 1/2 pink, and 1/4 white. All right? And so that's a little bit of a deviation, but it's pretty easy to see and to deal with. Okay, so here's an example of codominance, and this is an example in a lentil plant. Look at the seeds, and you find that the seeds can either have large black spots or small black spots. So this is called spotted, and this is called dotted. Okay? But it turns out that they're alleles of the same gene. Okay? Again, we're just going to call it a color gene, a C gene. And so if we make the F1s, right, again, genotypically they're all the same. They're CS, CD. But what they have is they have both black spots and black dots at the same time. Okay? So the two are codominant. You see the effect of each allele. Okay? So you see spots and you see dots. Okay? And then again, you should be able to calculate what happens if we go to the F2. So now you're going to get 1/4 will be spotted, 1/4 will be dotted, and 1/2 will be both spotted and dotted at the same time. All right? And that's where, again, you have two alleles of that, but they're codominant. So the genotypes show up in the phenotypes very clearly as separate things that you can see. All right. So a couple things to remember about dominant recessive relationship. It's not that the dominant allele, it doesn't subdue the recessive allele in any way. They don't interact in that kind of way. The thing to think about is that the alleles, right? They're just variations in the DNA sequence. And so what can happen? So we've talked about how you have a gene that's a DNA sequence. It's transcribed as an RNA made into a protein. We're gonna, as we go through the course, we'll give you more details of how that works. But you can imagine, for example, that you have an enzyme, right, that's catalyzing a reaction to synthesize a red pigment. And so the wild type allele, the normal function, has that enzyme. And it's giving rise to red pigment. And then if you have a mutation, that mutation could completely disable the enzyme. So it'll mutate a critical nucleotide that encodes part of the protein. The protein doesn't function, and now the thing is white instead, right? Because it doesn't make that pigment. And so then it makes sense also that the heterozygous in that case would show a simple dominance recessive relationship, because what's happening is if there's one normal copy of the gene, that makes plenty of enzyme and you still get a red flower. Okay? So when you think about it, as you go through some of the examples, we'll get into slightly more sophisticated examples, you can keep that in mind. Okay? And the character of dominant and recessive also depends on the level that we look at it. So if we look at it at a phenotype, we can characterize it at one level of being dominant and recessive. But if we're able to actually look at the molecules being produced or something like that, we may consider it differently in terms of dominance and recessive. Again, we'll get to some examples in a little bit. Okay? And then you might think that dominant alleles were more frequent, but that's not actually true, right? And the reasons for that can get kind of complicated, but you want to keep this in mind. So for example, there's a dominant mutation that causes extra fingers, okay, in humans, but it's actually very rare. So it doesn't dominate the population in any way. Okay? So it's a dominant allele, but don't think of that as then overriding everything and showing up in the entire population for some reason. Okay? Just keep that in mind. Okay, so that allele is unusual and it's dominant, okay, but the recessive trait is far more prevalent. Okay, so most babies don't have that; they have the right number of fingers. Okay. You also get situations where you have multiple alleles for a single gene. Okay, and a very good example, the one that's always talked about in textbooks and everything, is the case of human blood types. Okay, so they're the different blood groups are A, B, and O. Okay, and so people can either be A, B, O, or AB. Okay, and the reason for this is that there is a single gene called the I gene, and it has allele A, allele B, and then a lowercase I. And what this gene does is it encodes an enzyme, okay, that adds a carbohydrate group onto proteins on the blood cell, okay? And so it adds the A carbohydrate when it's the A allele, it adds the B carbohydrate when it's the B allele, okay? And the little I enzyme adds neither, okay? So that's why it's called O, okay, nothing. So then this is what happens. So if you have, the A allele, you add the A carbohydrate, the B allele, you add the B carbohydrate, the I allele, you add none. So then, because you have two copies of this gene, right? So if you're big A, or big I, then the only carbohydrate you have on your blood cells is A. If you're big B, or big BI, then the only carbohydrate you have is the B form. And if you're both A and B, then you have both carbohydrates on your blood cells. And if you're II, then you have neither. Okay? So that's how you get the 4 different blood groups. And so this is an example then where you have more than 2 alleles for a gene. Okay? And then you can see that. So in a sense, you can think of this as codominance, right? You see both of those effects of both alleles on that. Okay? So that's a good example where there are multiple alleles of one gene. Then you also get the effect of pleiotropy. So we're giving you examples where like a gene has a very specific phenotype. But in reality, often you get multiple things that go wrong. And so you have multiple phenotypes at the same time. And we'll go into these a little bit more, but examples include mutations that give you cystic fibrosis or sickle cell disease. There's actually multiple phenotypes that you'll suffer from, okay? But they're all caused by the same gene. So that's called pleiotropic, when a gene seems to actually have many functions and give many phenotypes. Okay? All right, and so then we can also see that sometimes a trait is actually acted on by 2 or more genes. In reality, a trait is often acted on by many genes, okay? They have some effect. And so that makes the process of deciding the phenotype in cross is a little more complicated. Okay? So because the genes can interact in various ways, and this is called epistasis, okay, how the genes interact with one another. And we'll give you some examples here. Okay? So in this case, so epistasis means that a gene at one locus alters the phenotypic expression of a gene at a second locus. Okay? So here's a real example. In Labrador retrievers, okay, coat color depends on 2 genes. Okay? And one of these determines the pigment color. Okay? So either black or brown. So black is dominant over brown. Okay? And then the other gene has allele C and big C and little c, and that actually determines whether the pigment actually gets deposited at all in the hair. Okay? So if you're big B, big C, you have black. But if you're big B, little c, then you actually don't have any color, okay, or you're tan, actually, very light color. Okay? And that's because you can make the pigment, but you can't actually deposit it, okay, so you don't get any color. And so this locus here, C, right, is able to have an effect on the phenotype that you see from the other locus. So you can write that out, okay? If you do a cross, if you have dogs that are heterozygous for both loci, Okay, and then you get your usual, Punnett squared, and you get the 16 here. Okay, but if you go through this, okay, so these ones, these 4, 3, 7, 8, 9, okay, they are all black. Okay, these three here are brown because they're homozygous for little b. Okay, but they have at least one big E allele. Okay, and so they're brown. But then these four are all white or yellow, very light colored. And that's because it doesn't matter what allele they have at B, whether they have big B or little b. The fact that they're homozygous for little e means that they can't deposit any of the pigment that they make, and so they're tan. So remember in the very simple dihybrid cross, we got that phenotypic ratio of 9 to 3 to 3 to 1. Okay? But instead here we get 9 to 3 to 4. And the reason is because now if you're homozygous for little e, it doesn't matter what you have at the other locus, right? You're just this light color instead. Okay? And so that's an example where you don't get the expected phenotypic ratio. But you've actually, of course, it's still a Mendelian pattern. You still have the right genotypes. They're showing up in the right ratios. It's just the phenotypes look different. Okay? And so that gives you a slightly different ratio. Here's another example. Let's say that you had some other flower, something other than peas, okay? And you had two white lines. So you had two true breeding populations of plants that gave white flowers all the time. Okay? And you kept them, let's say it was two different people doing this experiment in two different places. And they both had white flowered plants. But then they exchanged seeds when they crossed them, and they now suddenly got back colored flowers, red flowers. Okay? So why could that be? What's that? So they are, it's fine. You can think of them as both, they're recessive mutants, right? But why, when you cross them together, do they now suddenly give color? It's heterozygous. I mean, these have to be homozygous for whatever they're at, right? The locus they're at. Yeah, so there can be two genes that give the same phenotype, but they're two different genes, right? So you can think of it this way, okay? So here's like a little bit more concrete way to think about it. We're just going to make up something that explains this for the moment. So imagine that you have a colorless precursor molecule, and enzyme A acts on it. It makes it into another molecule that's still colorless, but then enzyme B can act on it to make it purple, okay, or red, whatever that color was. So now if you think about it, so if you're homozygous for big A and little b, or sorry, big A and big B, it means the pathway goes all the way through, and you're purple, okay? And so that would be true for all four of these, okay? But now you see what happens here. So now if I'm homozygous mutant for either A, mutant for little a here, or little b, those are white. Okay, as well as being mutant for both. Okay, so these are white. So what it could be is that one person actually had a mutation in A that gave them white flowers, and the other person had a mutation in B that gave them white flowers. Okay, but they're two different genes. They're two different enzymes in this pathway, the way we've drawn it. Okay, but can you understand why they both give white? Okay, because they both stop the pathway before the molecule has color. Okay, and so then the reason then that you get, so that's why that happens. So now let's imagine you do this. So now I'm going to take this flower, I'm going to take a plant that's big A, big B, little b, and I'm going to cross it again to itself. Okay, and I get 100 progeny. Okay, so how many of those are going to be purple, and how many of those will be white? Okay, do you want to spend a minute trying to do that? What's that? Well, think about it. So just that pathway that I drew. Okay, I'm just going to go ahead and show you so you can see how to work it out. Okay, so again, the idea is that you would have these different possible genotypes, right, from these combinations, and what you see is that these 9 would be purple, okay, because they have both at least one functional A and one functional B, meaning they're the phenotype A dash and B dash, right? The rest of these would be white. So for example, here it's got two wild type or big A alleles, but because it's little b, the pathway stops and you never get the purple pigment. Likewise here, it also stops because even though you've got homozygous for big B, you're homozygous also for little a. And then all of these combinations stop as well and give you white because at least at one locus, you're homozygous mutant with a small allele. Okay, so now you would get a 9 to 7 ratio. Okay, so about 56 purple to 44 white, something like that. Okay, so again, see, it looks odd to you. You do this cross and you get a ratio that seems to make no sense. So in a way, Mendel was also very lucky that he didn't have a lot of this happening because it would have been very confusing looking results. Okay, but we can understand this now as still being simple Mendelian inheritance patterns. Okay, so here's one more that I'm going to leave you with. And I'll leave you with this one, and then we'll do it as a clicker as soon as we start on Monday. Okay, so imagine that we have now a colorless precursor. Okay, an enzyme X acts on that, and it gives a brown product. Okay, and then enzyme Y acts on it, and it gives a red pigment. Okay, so if we think about that, then okay, let's now cross a wide-eyed animal. So let's think of these as Ys, maybe. Okay, so we can cross a white-eyed individual to a brown-eyed individual, which means we're crossing big X, little y, little y. Okay, because that would give you that would be in this case that's what the white eyes are. Okay, so this enzyme's not there. Sorry, that's going to give brown. And then if we're homozygous mutant to X, that's going to give white. Okay, does that make sense? Okay, so this is actually the brown one. This is the white one. Okay. So then the question is, what eye colors and what proportions do you see in the next generation? And to make it easier, we'll just narrow it down to these 4 right away. Okay, so what we'll do is go ahead and just leave you with that. You can work on that. I'm also going to post up onto VSpace some more sample problems, okay, that obviously aren't required, but if you start doing it, it'll help you.